Welcome back, everybody, to another deep dive. And today we're going to be doing something really cool. I think we're going cellular.
Oh, wow.
We're diving into the world of molecular biology.
Sounds complicated.
Well, it is, but it's also super fascinating.
Okay, I'm intrigued.
So basically, you sent us this lecture handout.
Yeah.
And it's all about how genetic information flows from DNA to RNA to protein.
That's right.
It's basically like the secret code of life.
Yeah.
How does a cell actually build itself? How does it know what to do?
Right.
And so we're going to try to crack that code today.
I love it.
So where do we even start with something like this?
Well, the best place to start, I think, is with the central dogma of molecular biology.
The central dogma.
Sounds very serious.
It does sound kind of serious.
Yeah, well, it's basically the fundamental principle
Okay.
that explains how information that's stored in DNA gets turned into the proteins that make up, well, everything.
Got it.
Yeah.
So it's like the master blueprint.
It is.
For building an organism.
Exactly.
So DNA is like the master library.
Think of it that way.
Holding all the genetic blueprints.
Yeah, and then those blueprints get transcribed into RNA,
Okay.
which acts as like a messenger,
Okay.
carrying the instructions to the construction site where proteins are made.
Ah.
You see?
So the construction site is the ribosome.
That's right. The ribosome is where it all happens.
Okay, cool.
Now, this process is universal.
Okay.
It happens in all living things.
Got it.
But there are some interesting variations between simple organisms like bacteria
Right.
and more complex ones like us.
So that's why this handout, BTCH 4251, LEC 1 and Lab 1, Molecular Biology Review.pdf.
Oh, you remember the whole thing?
I tried.
Yeah.
It highlights these differences with that diagram figure 1.1.
Right.
Shows how things work in prokaryotes versus eukaryotes.
That's it. Exactly.
Okay, so prokaryotes.
Prokaryotes are kind of like streamlined factories
Okay,
where things happen very efficiently.
got it.
Simultaneously. But eukaryotes, their separate nucleus,
Right.
they have a more compartmentalized approach.
Okay, so it's like a factory with dedicated departments.
A great analogy.
Yeah, I'm starting to see that city analogy coming to life.
I like it.
So let's zoom into one of these departments.
Okay.
Transcription.
Okay.
That's where DNA gets copied into RNA, right?
Precisely.
Okay. So imagine a team of specialized scribes in the nucleus,
I like it.
carefully copying those DNA blueprints into RNA messages.
I can see it happening.
Right.
Yeah.
And this scribing happens in stages?
Yes. 3 main stages.
Okay.
Initiation, elongation, and termination.
Okay, let's break those down.
Sure. So first, the scribing team needs to find the right starting point on the DNA.
Okay, so they need like a start signal, like a
Yeah.
big neon sign saying begin here.
Yeah, like a big flashing light.
Right.
And that's the promoter region on the DNA.
Okay,
It's where the enzyme that's responsible for doing the copying,
got it.
RNA polymerase,
Okay.
that's where it binds
So,
and gets ready to go.
so the promoter is like the address on the blueprint.
Yes.
It tells the scribes where to start copying.
A perfect analogy.
Awesome.
Now, during elongation,
Okay.
the scribes, they meticulously copy that DNA sequence into a growing RNA molecule,
So they're like building a chain
Yeah.
link by link
link by link.
based on that DNA template.
Exactly.
Got it.
And finally, termination.
Okay.
That's like reaching the end sign on the blueprint.
Got it.
RNA polymerase knows to stop copying,
Okay.
and it releases the completed RNA transcript.
Okay, that makes total sense.
Good.
But you mentioned earlier that eukaryotic transcription is a bit more complex.
Right.
So what makes it different?
Well, in eukaryotes, it's not just about copying that entire DNA sequence.
Okay.
There's an extra layer of editing involved.
Editing.
Editing,
You're
yeah,
cutting and pasting parts of the RNA message.
exactly.
Oh, wow.
So remember those non-coding regions in eukaryotic genes?
Yeah, the introns.
The introns, right?
Yeah.
They're like those unnecessary scenes in a movie
Right.
that need to be cut out
You gotta cut them out.
before it's ready for release.
Right.
Yeah.
In this movie, editing happens during RNA splicing,
Does.
right? And figure 1.2 in the handout
Right.
shows how different the final RNA is compared to that initial copy.
That's a great point. Yeah.
Yeah.
It really highlights that.
Yeah.
Yeah.
So who are the editors in this cellular movie studio?
Well, they're called spliceosomes.
Spliceosomes.
Spliceosomes, yeah.
Okay.
Incredibly complex structures made of RNA and protein.
Wow.
And they act like molecular scissors and glue,
Wow, that's so cool.
snipping out those introns and then splicing together the important parts, the exons.
Wow, so much is happening,
Um,
even before it gets translated into a protein.
I know, it's amazing. isn't it?
Yeah, figure 1.6 gives us a closer look at these spliceosomes in action.
Yeah.
It's mind-boggling.
It is mind-bogglingly complex.
Yeah.
But here's a fascinating twist.
Okay.
Some RNA molecules in those spliceosomes can act as enzymes.
What?
Now, we usually think of enzymes as being proteins,
Right.
but these RNA enzymes are called ribozymes.
Ribozymes.
Yeah.
Okay.
And they show us that RNA is more than just a passive messenger.
Wow. So RNA can be both the blueprint and part of the construction crew.
That's right.
That's amazing.
Now, this splicing process isn't always so straightforward either.
Oh.
Sometimes different combinations of those exons
Right.
can be spliced together,
Okay.
creating different versions of the protein.
From the same gene?
From the same gene. That's right.
Oh, that's like having a Lego set
Yeah.
that can build different models.
It is.
That's so cool.
It adds a whole new level of flexibility and complexity
Yeah.
to gene expression.
That's incredible.
Yeah. And figure 1.7 in your handout gives an example of how this might work.
Cool.
Yeah.
So after all of this editing, the mature mRNA
Right.
is finally ready for its starring role
Yeah.
in translation.
Yeah.
The process of actually building the protein.
Yeah.
All right, I'm ready to follow this mRNA out of the editing room and onto the set.
Let's go. Okay, so picture this, the cytoplasm. It's like this bustling city.
Okay.
And the ribosomes, they're like the construction sites where proteins are made.
Okay, so our mRNA, it's fresh from its editing session.
Right.
It's arriving at the construction site with the final blueprints.
It is.
But how does like a string of nucleotides
Right.
get turned into a functional protein?
That's the question.
It's like trying to build a skyscraper from code,
Right.
written in a completely different language.
Yeah, it's a good analogy.
You know,
So that's where the genetic code comes in.
the genetic code.
It's like the Rosetta Stone of molecular biology.
Oh, wow.
It translates the language of mRNA into the language of proteins.
So it's like a universal dictionary.
Think of it that way,
Okay.
that everybody in the cell understands.
So every 3 nucleotide sequence on the mRNA,
Called a codon.
a codon,
Yeah.
codes for a specific amino acid,
That's right.
which is the building block of proteins. Exactly.
Okay, so that's what figure 1.8 in the handout is showing.
It is.
The whole genetic code.
The whole thing, yeah.
It's incredible that like pretty much every living thing uses the same code.
It's amazing, isn't it?
It is.
It really is, yeah.
But how does the ribosome actually read this code
Okay.
and know which amino acid to add?
Well, that's where tRNA comes in. Transfer RNA,
Okay.
acting like a little delivery truck,
Oh, cool.
bringing the right building materials to the construction site.
Got it.
So each tRNA carries a specific amino acid,
Okay.
and it has this special address tag
Right.
called an anticodon.
An anticodon.
Yeah.
It matches up with the corresponding codon
On the mRNA.
on the mRNA. That's
So
right.
it's like a matching game.
It is. It is.
The tRNA with the right anticodon
Yeah.
docks onto the mRNA.
Yep.
Delivers its amino acid cargo.
Exactly.
Okay.
And then the ribosome acts like the foreman,
Okay.
overseeing this incredibly precise assembly process.
It's like a perfectly choreographed dance.
It really is.
Can we break down the structure of the ribosome a bit more?
Sure.
Like, how does it actually facilitate this protein assembly?
So the ribosome is kind of like a 2-part machine
Okay.
with a large subunit and a small subunit.
Okay.
And they come together
Okay.
to create a platform for protein synthesis.
So they clamp onto the mRNA
Right.
and they create like a workspace
They do.
for the protein to be built.
Exactly.
Okay.
And within this workspace,
Okay.
there are 3 key binding sites for the tRNA molecules.
Okay, cool.
And you can actually see this in figure 1.10 in your handout.
Okay.
It illustrates these sites,
Okay.
the A site, the P site, and the E site.
Okay, so the tRNA carrying the next amino acid enters at the A site.
That's right. The A site is like the loading dock.
Okay.
So the tRNA with the matching anticodon binds there,
Okay.
bringing in the next building block.
Okay.
Then the growing protein chain, which is attached to a tRNA at the P site,
Right.
gets transferred over to the new amino acid.
So the P site is where the actual protein chain is being assembled.
You get it, think of it as the main assembly line.
Okay.
And once that transfer happens,
Okay.
the empty tRNA
Yeah.
exits the ribosome at the E site,
Okay.
like a truck leaving the construction site
Right.
after dropping off its cargo.
Okay.
You see?
It's amazing how this like tiny molecular machine
I know.
orchestrates such a complex process.
It's incredible, isn't it?
Yeah, and it all happens in stages, right?
It does.
Like initiation, elongation, termination.
the same pattern we saw before.
Right.
Yeah.
So like any well-coordinated project, protein synthesis has a clear beginning, middle, and end.
Exactly.
Okay, so let's start with initiation.
Okay.
How does the ribosome know where to begin building the protein?
Remember that start signal on the mRNA?
Yeah.
The start codon?
The start codon.
That's where the ribosome assembles on the mRNA.
Okay.
With a special initiator tRNA.
Okay.
Carrying the amino acid methionine.
Okay. So every protein starts with methionine.
It does, that's right.
That's fascinating.
Yep.
And figure 1.11 shows this initiation complex coming together.
It does.
It looks like a lot of moving parts.
It is a lot of moving parts.
Yeah.
But once that initiation complex is assembled,
Okay.
the real building begins during elongation.
Okay, so that's where amino acids are added one by one
One by one.
to the growing protein chain
Yes.
based on the instructions on the mRNA.
That's right.
Okay.
It's a cycle of codon recognition,
Okay.
peptide bond formation,
Got it.
and translocation.
Okay.
So the ribosome is moving along the mRNA like a train on a track,
Got it.
reading each codon and bringing in the matching tRNA.
And as each tRNA delivers its amino acid, it gets added to the growing protein chain.
Exactly like adding beads to a string.
Right.
Yeah.
That's a good one.
And you can see this beautifully illustrated in figure 1.12.
Okay.
It shows that elongation cycle.
Awesome. It's remarkable how quickly and accurately this all happens.
They're pretty amazing.
But eventually the protein has to be finished, right?
Of course it does.
So what signals the ribosome to stop adding amino acids?
Well, there are special stop codons on the mRNA
Okay.
that act like a red light
Got it.
at the end of the construction site.
So when the ribosome hits one of these stop codons,
Yeah,
it knows it's time to release the completed protein.
that's right.
Okay.
So a release factor protein binds to the stop codon,
Okay.
and that triggers the disassembly of the whole complex.
So the ribosome separates from the mRNA,
It does.
and the newly synthesized protein is set free.
Exactly. And figure 1.33 shows this final step.
Okay, so now we have a finished protein ready to do its job in the cell.
Well, not so fast.
Oh, there's more.
There's a bit more, yeah.
What kind of modifications are we talking about?
Well, for one thing, proteins need to fold
Okay.
into very specific three-dimensional shapes.
Right, because a protein's shape determines its job.
Exactly.
Okay.
And sometimes proteins, they need additional modifications too, like the addition of chemical groups or the removal of certain parts.
It's like tailoring a suit.
It is.
Is it perfectly?
Exactly right.
Okay.
And what about proteins that need to be sent to specific locations within the cell?
Right.
Yeah, well, some proteins have special signal sequences
Okay.
that act like address labels,
Oh, cool.
guiding them to their proper destinations.
So figure 1.14 shows how a signal sequence can target a protein to the endoplasmic reticulum.
It does, that's right.
Okay, cool. It's incredible how much complexity is packed into this process.
I know, it's amazing, isn't it?
Yeah, earlier we talked about the differences between prokaryotes and eukaryotes
Right.
in terms of transcription and translation.
Yeah.
Are there any other key distinctions that stand out?
There is one, yeah.
Okay.
Prokaryotes, they have this amazing ability
Okay.
to couple transcription and translation.
What does that mean?
It means that ribosomes can start translating an mRNA molecule
Okay.
even before transcription is finished.
Wait, so they're doing both processes at the same time?
They are.
That's not even possible.
Well, remember, prokaryotes don't have a nucleus
Right,
separating their DNA from the cytoplasm.
right.
So as soon as that mRNA starts being transcribed,
Okay.
ribosomes can latch onto it
Got it.
and begin translating.
It's like a super efficient assembly line.
It is very efficient.
Figure 1.15 shows this happening in a bacterial cell.
It does, yeah.
It's mind-blowing to realize that all of this intricate activity is happening inside every cell of our bodies constantly.
It's pretty amazing, isn't it?
We've covered so much ground already,
I know.
but we haven't even touched on DNA replication.
Yeah, we'll save that fascinating process for another deep dive.
Okay.
I think for now we've given you a good understanding of how genetic information flows
Right.
from DNA to RNA to protein.
I agree, but before we move on,
Sure.
I'm curious. Earlier, we talked about how alternative splicing
Right.
allows one gene to produce different proteins.
Yeah,
Are there any other exceptions to this central dogma?
that's a great question.
Is it always a one-way street from DNA to RNA to protein?
Well, the central dogma is a fundamental principle,
right.
but there are some fascinating exceptions.
Oh, cool.
For example, some viruses,
Okay.
they have RNA genomes
Instead of DNA genes.
instead of DNA genomes.
If they like bypass the DNA to RNA transcription step altogether.
They do. They go straight from RNA to protein.
Wow.
And some RNA viruses, like HIV,
Okay.
have an enzyme called reverse transcriptase.
Reverse transcriptase.
Yeah, can actually transcribe RNA back into DNA.
Whoa, that's like reversing the flow of information.
It is. It is.
It's amazing how some organisms can bend the rules of molecular biology.
It's pretty remarkable.
Okay, so we've explored the central dogma. the fascinating world of protein synthesis,
We have.
and even some mind-bending exceptions to the rule.
We did.
What are some key takeaways you hope our listener will remember from this deep dive?
Well, first and foremost, I hope they gain a deeper appreciation for the elegance and complexity of life at the molecular level. It's truly awe-inspiring how these tiny molecules orchestrate the processes that make life possible.
I totally agree. It's like peeking behind the curtain
Yeah,
and seeing the intricate machinery that keeps everything running.
that's a great way to put it.
And understanding these fundamental processes opens up so many possibilities in fields like medicine, agriculture, and biotechnology.
It does.
So by understanding the secret code of life, we can potentially unlock solutions to some of humanity's biggest challenges.
Absolutely.
That's incredible.
And perhaps most importantly,
Yeah,
this deep dive highlights the importance of continuous learning and exploration.
right.
There's always more to discover in the world of molecular biology.
So well said. Okay, before we wrap up this incredible journey into the cell,
Yeah,
is there one final thought-provoking question
I do.
you'd like to leave our listener with?
Given what we've learned about the intricacies of gene expression,
Yeah.
it's worth pondering this.
Okay.
How might our understanding of these processes shape the future of medicine and healthcare?
That's a great question to consider. It seems like we're on the verge of a new era of personalized medicine,
Right.
where treatments can be tailored to an individual's unique genetic makeup.
Exactly.
It's both exciting and a little bit daunting to think about the possibilities.
I agree.
Yeah, it really is amazing to think about just how far we've come in understanding like these fundamental building blocks of life.
And we're only just scratching the surface.
I know there's so much more to learn.
Yeah.
Okay, so before we wrap up this incredible deep dive into molecular biology,
Yeah.
let's do a quick recap for our listener.
Sounds good.
So we started our journey with the central dogma,
Right.
that elegant principle that explains the flow of genetic information from DNA to RNA to protein.
It's a beautiful concept.
Yeah.
Yeah.
We then zoomed in on the intricate processes of transcription and translation,
Yes.
marveling at the molecular machinery like RNA, polymerase, spliceosomes, and ribosomes that make it all happen.
Those tiny little machines are incredible.
Yeah. We also explored those fascinating post-transcriptional and post-translational modifications,
Right.
highlighting how RNA and proteins are like fine-tuned to perform their specific task.
I think they have their own little finishing school.
Exactly.
Yeah.
And we even touched on DNA replication, that essential process for cell division and the continuity of life.
It's the foundation of it all.
Yeah.
Yeah.
We discussed some practical applications of this knowledge, from developing like life-saving antibiotics to the revolutionary potential of gene editing with CRISPR.
It's amazing how understanding these processes can lead to such incredible advancements.
Yeah, and as we explored these powerful tools, we were reminded of the importance of considering the ethical implications
Right.
of manipulating these fundamental processes.
Absolutely. It's a reminder that with great knowledge comes great responsibility.
Absolutely. So well said overall.
Yeah,
I hope this deep dive has left our listener with a sense of wonder
I hope so.
and a deeper understanding of the intricate mechanisms that underpin life itself.
It's a fascinating world down there.
It really is. I couldn't agree more. It's been an incredible journey exploring the cellular world.
It has.
And I have a feeling this is just the beginning of our molecular adventures.
I think you're right.
There's so much more to discover.
A whole universe of knowledge waiting to be explored.
Exactly. So until our next deep dive,
Okay.
keep exploring, keep questioning, and never lose that thirst for knowledge.
I love it.